All right, so in this last section of part 3, we're going to finish off looking at the different types of things that can influence your analysis of an analyte. So we got through the sensitivity and the selectivity in the last lecture. And in this lecture, we're going to focus starting on the robustness and the ruggedness of your methodology. So if your method is relatively free from chemical interferences, it can be applied to the determination of analytes in a wide variety of sample matrices. These methods are said to be robust, right? So disregarding what type of sample matrices you have your analyte in or mixture, if your methodology can handle that, then it's going to be a robust methodology. If you have a rugged method, this is in reference to how insensitive your method is to changes in the experimental conditions. So things like acidity, temperature, or reaction time, your method is rugged if you can change a lot of these features and still be able to analyze your analyte in a reproducible fashion. So the next consideration that we need to have is the scale of the operation. So when you're considering the scale when choosing your method, there are 3 limitations. So this includes the amount of sample that you have available for analysis. Then you need to think about the concentration of your analyte, what you really want to find within your sample, and then the absolute amount of analyte that's needed to obtain a measurable signal. So here on this chart over here, you can see that this is a log scale. So you're looking at the analyte as a percent of your sample by weight. And so if it's a major constituent of the sample, it will be between 1% and 100% of the sample. If it's a minor constituent, it's going to be between 1/100th and 1%. If it's a trace sample, this goes all the way down into the parts per billion. And then you can have ultra trace as well. So if your analyte only makes up a tiny, tiny, tiny percentage of your sample, this would be an ultra trace sample, and you would have to have very special methodology that could be able to detect sample in that range. So on the x-axis of the scale, this is a measure of how much sample that you actually have. So if you have a lot of sample available, that's going to be in the range of 100 milligrams to 1 gram is a macro scale. 10 milligrams to 100 milligrams is on the meso scale. If you're down at 100 micrograms to 10 milligrams, that's going to be the micro scale. And then anything really below 100 micrograms is on the ultra micro scale. So you can see if you have very little sample and then it also only contains a very small percentage of your analyte, then your ability to be able to detect that analyte with your methodology becomes very difficult. So for the samples, your scales are macro, meso, micro, and ultramicro. And for the analytes, they can be major constituents, minor constituents, they can be in trace amounts, or they can be in ultra trace amounts within your sample. So you have to consider both of these features when you're deciding on what methodology you want to use to analyze your samples. So consider this hypothetical total analysis method. So in this situation, the minimum detectable signal requires 100 milligrams of analyte. And if the analyte is a minor analyte with a concentration of 0.1% weight by weight, how much sample do you need to be able to detect the analyte? So I would stop the video at this point and try to do this question and see what you come up with. And we will answer this in class. So other things to think about when you're setting up your methodology: what equipment is going to be needed? Do you have that available to you? How much time does the analysis take? And what is the cost of that analysis? Right? All of these features are going to help you determine which methodology that you can actually do. Okay, so let's talk a little bit more about compensating for interferences within your samples. So potential interference may be present in the sample itself. It can also occur in the reagents that are used during the analysis. So how can we minimize these two sources of interference? So in the absence of any interferent, the total signal measured during the analysis, the S measured, is going to be a sum of the signal that's due to the analyte, and it's going to be due to the signal due to the reagents. So we can think about this as the measured signal is going to be a combination of the signal from the analyte and the signal from the reagent. So any background that you might have from the reagents. So for the total analysis method, we can substitute in the proportionality constant and the moles of the analyte for the signal here. And that was from our initial equation that we were doing. And for the concentration method, we can substitute in the proportionality constant and the concentration of the analyte. And then you still have to include the signal from the reagent. So when you have this situation here, what you would do to compensate for the signal of the reagent is to have a method blank. So if we have a method blank that does not contain any of the samples and we run it through our system and we get the signal, that's going to be the signal of the reagent. And then we can put that into our equation and subtract it off of the measured signal with our sample so that we can isolate the signal that's coming from the analyte. So method blanks are very, very common ways to get rid of signal that's caused by the reagents that we might be using. So if we have interference in the sample matrix rather than in the reagents, this is more difficult to get around because we can't just do a method blank, right? And we can't just take the analyte out of the sample itself and then see what that background measurement is going to be. Plus, if it's interacting directly with our analyte, it could be interfering in a negative way and reducing that signal as well. So this gets a little bit more complicated when you have an interferent that's also in the sample matrix. So when we're measuring this, then the signal that's measured is going to be a combination of all three things, right? It's going to be from the signal of the analyte. So we can put into that equation the proportionality constant for the analyte and the moles of that analyte. And then it's going to also be the sum then of the signal that's coming from the interferent that's in the sample matrix. So we can substitute in the proportionality constant for the interferent and the moles of the interferent, and then the signal that's coming from the reagents. So we always have to have that reagent blank to subtract out any signal that might be coming from the reagent. So if the identity and the concentration of the interferent is known, it can be added to the reagent blank, right? And then we can figure out how much it's coming from that, and we can get an idea of what this signal would be, and then we can subtract off that whole thing together. However, this often is not the case when you're working with real-world samples. We don't know what the interferent is, and we don't know the concentration of it. So if the interference identity or concentration are also unknown, it has to be added to our term into the equation. So we have to also include it here. All right, so there's two ways to deal with this situation if we don't know the concentration of an interferent inside our sample, or maybe we don't even know the identity of it. So to be able to do this, we have two methods that we can use. The first one is to solve for just the analyte. We would have to separate the sample further to purify the analyte away from the interferent prior to the analysis, right? So this may or may not be possible. And then we also have to take into account that anytime you do a separation step or a purification step, you're going to lose some of your sample. And so that's going to have to factor into your analysis as well. How much do you actually retain when you're doing that analysis? Otherwise, you can solve for this equation down here if you have two measurements that you've taken independently from one another. This is going to give two equations that can then be solved simultaneously for either the Na and Ni or the Ca and the Ci if you're using concentrations. So how does that exactly work? Well, let's take a look at an example. So here's our example problem. So we're going to analyze the concentrations of the two analytes A and B under two sets of conditions. So under condition 1, we have the calibration sensitivities for A and B that are given. So this is the sensitivities for or proportionality constants at calibration 1 or condition 1. And this is the proportionality constant or sensitivity constant for B under condition 1. And then we have condition 2 and condition 2 for both A and B. They also give us the signals measured at both 1 and 2. And then from this, we have to determine the concentration of A and B. And for our problem, we're assuming that the signal from the reagent is 0 under both conditions. If we were actually doing this in the lab, we would also have to do the method blank and then subtract off any signal from that reagent from our measurements before we could do this calculation. So then we need to write out our two equations with the values that we know. So you can see here we've got the signal for condition 1, and then we just put in the sensitivities or the constants in for A and for B. In this case, we've got concentrations. And then for sample 2 or condition 2, we have our values as well. So then we perform a step that will enable one of the terms to subtract out. And so we're going to pick Ca, and we're going to want to subtract equation set 1 from equation set 2, but we'll want the Ca term to be able to cancel out and be 0. So to do that, we have to get these equal to each other, right? So we need to get rid of the 76 and we need to get a 33 here. So to do that, if we multiply this term by 33 over 76, the 76's will cancel out and we'll get 33 there. So that's how we're going to do this. But if we do that to this term, we're going to have to do that to every term in our equation. So we distribute that out, and then it's just like multiplying everything by 1, right? So it's going to be that same proportion all the way through, and we do the math for that, and we end up getting our first equation is going to be 14.5 equals 33 per parts per million Ca, and then 80.8 per parts per million for Cb. And then we can subtract this equation from equation 2. So here we've set that up on this page here, and you can see here we'll get 15.2. This whole term is going to be 0, right? Because if you subtract this from this, and then if you subtract this from this, you'll get 162 per parts per million for Cb. We can then solve for Cb. Once we've solved for Cb, we can put that back into either equation and we can solve for Ca. All right, so this is another method that we can use to get rid of interference in the sample. All right, so we do need to know information about the interference. We need to be able to calculate the proportionality constants for these. Right? And so we would need to know the identity of the interferent in this case so that we could do some experimentation in the lab and figure out what these proportionality constants or the sensitivities are going to be. All right, so that's two ways that we can deal with the interference. Either purify your analyte away from the interferent, or if you know what your interferent is and you can calculate the proportionality constants or the sensitivities for that interferent, then you can go ahead and you can take two samples isolated under different conditions, and then you can subtract them out from one another to be able to solve for the concentration of A and concentration of B. So two other features of your methodology that are very important are calibration and standardization. So we're going to focus a lot on creating standards and using standards in Chapter 5. So that is up and coming. But let's just get a little bit of a definition for both of these. So calibration is making sure that your equipment or your instruments that are used to measure signals are operating correctly. And to do this, you're going to use a standard that's known to produce an exact signal. So you're calibrating your instrumentation or equipment. Standardization then is the process of experimentally determining the relationship between the signal and the amount of analyte, right? And this is the value of K, or the sensitivity, in the equations, right? So you need to be able to calculate your proportionality constant. And you do this through this process of standardization. So for the total analysis method, standardization is usually defined by the stoichiometry of the chemical reactions responsible for the signal, right? So we said this is going to be a known sum and it's not going to change, right? And so the chemical reaction is going to be the important component there. For the concentration method, however, remember that the relationship between the signal and the analyte's concentration is a theoretical function, and so you have to calculate it with the experimental measurements because the value of K is going to vary depending on the instrument that you use to make those measurements and on the concentration of your analyte. So these have to be done experimentally for the concentration method. So how do you determine K? So you need to do a standardizing for any sort of method. So you'll need to make some standards for your analysis. Ideally, those standards are going to be the analyte that you're interested in evaluating in your sample matrix from wherever you're collecting it, right? So if I want to, I don't know, analyze lead in water, for example, right? I'm going to have known concentrations of lead in my standard, right? And then I'm going to measure them using my instrument to get that signal for the lead concentration. Right, so on this diagram, it's showing you the readings that you might get out. So if this was increasing lead concentration, right, you can see that there is a dynamic range where the standard is giving a linear signal, right? Before, so there are specific features of this that you have to take into account when you're looking at a standard curve. Right? So here you can see that the curve starts deviating away from the linear range. So this is the high point up here that you can go to on this standard. If you're above this concentration, you'll notice that your instrumentation is no longer able to read these samples correctly. It's going to give you a false value. It's going to undershoot the actual real value. Because you're not getting in the linear range anymore out here. So if your sample from your sample matrix that you collect is higher concentration than your standard that you've run, you're going to have to dilute back your sample so that you get it into this dynamic range that you want to use for calculation. All right, so this upper limit here, this is the limit of linearity, or your LOL, right? And so this is as high as you can read your standard concentration. If you have a concentration of lead in our case that is higher than that, your instrument can no longer read it correctly or in the linear range, right? So you can't, if you get a value in your sample that's higher than that, it's not going to be accurately read. So you would have to say, oh, I need to dilute my sample and read it again until it's in this dynamic range. This LOQ is the limit of quantification. It's the concentration at which the quantitative results can be reported with a high degree of confidence. So it is a limit where you have the lowest amount of analyte in the sample that can be quantitatively determined with suitable precision and accuracy, right? So we're going to define this in our medium range, right? Within 1 to 5% accuracy. That would be our limit of quantitation. Below this is our limit of detection, our LOD. So we can detect that the analyte is present, but we're not getting real accurate range down here. And our variability is above 5% in this range. And so we can't use this area of the curve either. So for each type of analyte that you're trying to analyze, you need to have some sort of standardizing method that you're using to be able to compare your analytes with, right? And help you determine the quantity of analyte that's actually in your sample. So this is known as a calibration curve. And you'll be doing these in the lab as well. And in fact, chapter 5 really focuses on the different types of standardizations that you can do. And so we'll spend some additional time talking about these in more detail. So proper sampling is also needed when you're taking samples out in nature or from a group. You need to ensure that the samples are representative of the population that you're sampling from. So you want to get a representative population in there. So we will come back to this topic as well later in the term and look at different types of sampling methods that can help ensure that we get an accurate representation of any population that we might be interested in studying. All right, and our last main consideration is validation. So validation is the evaluation of whether the precision and accuracy obtained by following the procedure are appropriate for the problem we're trying to solve. Validation also ensures that written procedures have sufficient detail so that different analysts or laboratories that are following that procedure can obtain comparable results. And validation is usually done with a standard that is very similar to the sample being studied and must be done with replicate samples, usually 3, to ensure precision and accuracy. So there may be an internal standard that's processed exactly like the samples and compared with the standard curve. So validation, right, has to be done in addition to that standard curve. So besides all these considerations that we're taking into account now when we're designing our procedure, a protocol also contains very explicit instructions regarding internal and external quality assurance and quality control. These are often abbreviated as QA and QC for the procedures, right? So if you have a procedure that you want to amplify up into a protocol that might be adopted by like the EPA, you have to have these quality assurance and quality control measures in there. So you have 2 ways to do this, the internal and the external. So for internal quality control, this is going to include steps that are taken to ensure that the analytical work in a given laboratory is both accurate and precise. And for the external quality control, this is going to usually involve a process in which the laboratory is certified by an external agency. So maybe the FDA comes in and looks at things, or maybe the EPA would come in and look at how things are being analyzed, and they okay the process. Okay, so if you're actually running a protocol, like if you had an EPA contract laboratory program, for example, you would have to go through a long series of steps to be able to run samples that could be processed and analyzed for the EPA. So this would include your start. You'd have to do your standardization, and then you have to do a check, right? So you have your calibration curve is then verified using an internal calibration verification, which is called an IVC, and then it's compared with the blank, the ICB. So if these are both okay, then you can go on. If they're not, you have to identify the problem with the methodology or the reagents or the instrumentation. Do the standardization again and do the check again. Make sure the instrument is working correctly. Then you can go on. So for this, the IVC, that internal control, has to fall within 10% of the expected concentration. Otherwise, the analysis of samples has to stop and you have to address and correct the problem wherever it's coming from. Standardization then is re-verified every 10 samples, continuing calibration verification and the calibration blank. So if they're in sample range, then the analysis can continue. If they're not in range, then you have to discard your last set of 10 results. You have to figure out where the problem is at, and then you have to start again. So it continues this way until all the samples have been processed. All right, so protocols are a lot more in-depth and have a lot more quality assurance steps that are required when you're doing this for measurements where precision and accuracy have to be very highly maintained. All right, and that is going to end chapter 3 for us. And there are some keywords that you should go back and make sure that you are familiar with and know coming from this chapter. All right.